With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello students, have a look at the question. Question is about the assertion reason. Assertion is the inactivation of any vector gene due to insertion of desired gene called as a insertional inactivation. Reason is sometimes restriction sites are also present in between the gene sequence of vector. Options, we have to find whether assertion reason true or false, and if it is true, reason is correct explanation of assertion. So if we'll see the insertional inactivation, so here we will understand the insertional inactivation. Insertional inactivation means, as the name indicate, when any we can say that when any gene is when any foreign gene, desired gene, when any desired gene that is inserted into the gene sequence of a vector, gene sequence of particular gene sequence of vector, so that gene it get inactivated, because insertion of that gene into the gene sequence of in vector, that will result in the inactivation, inactivation of gene, because that gene will not remain intact. So that is known as the insertional inactivation. We will better understand it by taking an example of the plasmid, that is PBr322. PBr322, it is a common widely used plasmid. It has two selectable marker, that is it is having tetracycline resistance, other is it is having the ampicillin resistance. So here we can see that the tetracycline resistance and ampicillin resistance. So when any desired gene that is being inserted into the tetracycline resistance. So here this is a particular yellow highlighted is a gene tetracycline resistance. Here desired gene that has been inserted. It means this tetracycline resistant gene that will not remain the intact, and the recombinants where the gene has been inserted, they will not show resistance toward the tetracycline, although they will show the ampicillin resistant. But on the other hand, non-recombinant in which DNA hasn't been inserted, because so that's why they will show the resistance toward the tetracycline. In this way, it acts as one of the selectable marker. So this is the selectable marker, and this insertional inactivation, it acts as one of the method to distinguish between the recombinants and the non-recombinants. It can choose between the recombinants and the non-recombinants. So this is widely used method to distinguish the recombinants and recombinants. Now, how this gene sequence will be added into the vector? This is by only if there is any restriction site is available. If any restriction site is available within that gene sequence. Because if it will not be available, then there will be no cut and no addition of the new sequence is available. So then only this will be insertional inactivation that can work. Now if we'll see the assertion is inactivation of any vector gene due to insertion of desired gene is called insertional inactivation. Yes, this is called as the insertional inactivation. Sometimes restriction sites are also present in between the gene sequence of vector. Yes, this is correct. That's why only it is feasible, because if restriction site will not be present, how we can cut this gene sequence, and then how it will be insertion is possible. So both assertion reason, two reason is correct explanation of assertion. So here this is correct, rest 3 are incorrect option. Option A is correct. Hope you get it. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.